Here we're going to look at the balance in hearing sensory receptors. Starting with hearing, but not from a human standpoint, but from a fish's standpoint. Fish have a lateral line, and this is their sense of hearing. They're able to sense objects that reflect pressure waves in low frequency vibrations. This red line shows where they might be located in this shark. We see this distinctive line here on this fish. Now on this fish, the striped bass, you might initially think there's multiple lateral lines, but there's technically only one. If you look very closely at this line here, the darker line is actually the lateral line. So this fish only has one lateral line despite many stripes. This is a striped bass caught in the Connecticut River after launching from the Rocky Hill boat ramp. Now continuing on, in fish, under their skin, they have these canals that have sensory structures containing hair cells projecting into a gelatinous cochlea. These vibrations produce movements, and hair cells bend and depolarize associated with sensory neurons. So we see here the evidence of this moving back and forth, and you see how it's all kind of attached to all these different nerve fibers. This is how the fish is able to sense these pressure vibrations through the movement of this cochlea. And a little bit more intensive an image here, we see how it's kind of organized in all these different sensing either hair cells. It's this movement back and forth of these low pressure waves that's being interpreted by the fish, and that's their sense of hearing without having a distinctive ear like we do. Now in humans, sensation of gravity and motion. So you may be wondering why this is located here when we're talking about hearing and ears. Well, it's very important because your sense of balance is related to your inner ear. So there's receptors in the ear that inform the brain where the body is in 3 dimensions. Gravity is detected by the shifting otolith sensory cells, and these are located in a gelatin-like matrix, and they're composed of your inner ear. So the force of gravity is what's causing them to move. Tilt your head forward, gravity is pushing them all down, and that's your sense of where you are in 3-dimensional space. So your ability to walk as a youngster is actually dependent on development of your inner ear, so you're able to sense 3 dimensions, so that it helps you be able to maintain your balance, and this is the reason why. The motion, so balance is like motion. If you tilt your head forward, the force of gravity moves. Well, motion is detected by the deflection of these hair cells by fluid in a direction opposite of that motion. So that acceleration standpoint, they're going to go the opposite way. These hair cells are found in the cochlea, and they're tent-like assemblies in 3 semicircular canals. So a very similar structure to the fish, also using sensory receptors in hairs. Here we're just using it now to detect motion, and this is kind of related here of acceleration at rest or deceleration is all sense of motion based on the movement of these hair cells in 3-dimensional space and your body's perception of those. Now, properties of sound. Sound is simply a pressure disturbance, altering areas of high and low pressure originating from a vibrating object. Here we see a tuning fork, composed of areas of refraction and compression, represented by a sine wave here, in wavelength, which is the distance between 2 consecutive waves, either 2 consecutive peaks here, or it would be 2 consecutive troughs. A frequency is the number of waves that pass a point per unit of time, and the amplitude here is the distance between the origin and the crest, or it could be the origin and the trough. That would be the amplitude. And the speed is simply wavelength times frequency. Now, frequency is the number of waves that pass through a given point at a given time, and pitch is the perception of different frequencies. We hear basically from 20 to, if you're lucky, 20,000 hertz. And it's determined by different frequencies causing different parts of the membrane to vibrate. Different sensory neurons are fired, and this is how you're able to determine different pitches. Now, amplitude is the intensity of the sound measured in dBs, the decibels, and loudness is subjective interpretation of the sound intensity. Relates to how often neurons fire. So for sound waves, wave corresponds to pitch. Amplitude of the wave corresponds to the volume. The sound wave shown with the dashed line is softer in volume than the sound wave shown with the solid line. So here we're looking at our volume in comparison. So you notice it's basically the same pitch, same kind of different amplitude, same wavelength, I should say. Our different amplitudes here, but our wavelengths remain the same. And we see here the kind of small talking quiet bird. We see the very angry large bird here, indicating that difference in our amplitude, even though our wavelengths are actually remaining the same. Continuing on, when sound is heard, air vibrations detected. It's detected in your eardrum. Your eardrum is a membrane that's pushed in and out by waves of pressure. And there's 3 small bones. Ossicles are the bones that are located on the other side of the eardrum and increase with the vibration force. Amplified vibration is transferred to the fluid within the inner ear, and the inner ear chamber is shaped tightly coiled snail shell called the cochlea. We see that here in purple. We're going to investigate a little bit further. So we're transferring those sound pressure waves to our eardrum, ultimately to the bones located here. Now in the cochlea, there are hair cells that rest on the membrane running up and down the chamber, and they're converted by other membranes. We see that here. This is a cochlea here, the region that's coiled like a snail's shell. Sound waves entering the cochlea cause the membrane to sandwich and vibrate. Bent hair cells send nerve impulses to the brain. It's kind of like all these like receptors here. Our nerve basically movement is how we're able to interpret those different pressure waves located in the air. This is found through transduction, which are vibrations that travel up the fluid-filled interior of the cochlea. Different thicknesses of the membrane vibrate in response to different frequencies of sound. We kind of see that evident here. Here's 7,000 hertz, 5,000, 4,000, 3,000, 2,000 hertz, 20,000 located right here. Not heard by very many. Again, the same principle applies here with our hair cells. And the cross-section of the cochlea here, the top right figure, note that there's the addition of the upper canal and lower canal. The cochlea also has a middle canal. So here's our upper, here's our lower, and here's our middle canal. So we're zooming in here. The bottom image is the site of the sound actual transduction, and this movement of the stereocilia and hair cells results in the action of potentials that travel along the auditory nerve. That's ultimately being sent to the brain, and that's how we are able to perceive sound.